Picture this: boiling acid pools, frigid Antarctic deserts, crushing deep-sea trenches, and radioactive wastelands. Sounds like a list of places where nothing should be alive, right? Wrong. Turns out there are living things that love this stuff. They're called extremophiles. These little creatures thrive in places that would instantly kill most life on Earth. But what exactly is an extremophile? The word comes from the Latin extremis, meaning extreme, and Greek philia, meaning love. So, an extremophile is literally an extreme lover, an organism that loves extreme environments. In biological terms, extremophiles are usually microorganisms, life forms so small you need a microscope to see them. Most belong to the ancient domains of life, bacteria and archaea, which have been evolving for billions of years in some of the most inhospitable corners of the planet. Bacteria and archaea are both simple, single-celled organisms without a nucleus, but they differ in their cell structures and genetics. Archaea have unique membranes and enzymes that let them survive extreme environments, whereas bacteria are more common in everyday life, from yogurt to infections. So what kind of extreme are we talking about? Extremophiles can be found in places like boiling hot springs above 100 Celsius, sub-zero environments well below freezing, salt flats with salt concentrations that would kill most cells, deep-sea trenches with crushing pressures, and even nuclear waste sites filled with deadly radiation. And here's the most interesting part. Many of these organisms don't just tolerate these conditions, they depend on them to live. In fact, if you brought them into a mild, room-temperatured lab, they might not even survive. Different extremophiles are adopted to different extremes. For example, thermophiles love heat, whereas halophiles are obsessed with salt, and radiophiles survive high levels of radiation. But out of all the extremophiles out there, one of them is my personal favorite, an organism so tough it's earned nicknames like Conan the Bacterium. Meet Deinococcus radiodurans, a polyextremophile, meaning it can handle multiple kinds of extreme environments. This microscopic marvel can survive drying out completely, freezing, acidity, high doses of ionizing radiation up to 5,000 grays. That's thousands of times the lethal dose for humans who can withstand 2 to 5 gray. It's even more than a tardigrade, the most well-known extremophile. It can even withstand exposure to the vacuum of outer space. Yes, NASA sent it to space and it lived. So, how does D. radiodurans pull this off? It comes down to DNA repair. Radiation, dehydration, and toxic chemicals all do the same thing to living cells. They break DNA apart. For most organisms, that's the end, but D. radiodurans can take massive DNA damage and bounce back like nothing happened. Why? Because it's got some of the most efficient DNA repair systems known to science. The key is a molecular repair process called homologous recombination. I'll explain it in 5 steps. Step 1. DNA takes damage. When D. radiodurans is exposed to something like high radiation, it causes double-strand breaks in the DNA. That simply means that both strands of the DNA helix are cut, not just one. In most organisms, just a few breaks is enough to cause chaos. Genes are lost and information is scrambled. But D. radiodurans might experience hundreds to thousands of these breaks and recover. How many broken DNA strands can your body handle? Probably not 1,000. Step 2. Backup copies to the rescue. Here's its secret weapon. D. radiodurans keeps multiple identical copies of its genome, 4 to 10 copies to be specific. So even if one copy is destroyed, there are more versions floating around in the same cell. Now, the cell just needs to find the matching DNA sequences between the broken piece and an intact copy. This is where the homologous in the homologous recombination comes in. Homologous just means that the DNA sequences are the same. It's like keeping multiple hard drives of the same file. If one crashes, no big deal, you've got backups. Step 3. Strand invasion. Proteins in the cell will scan the DNA fragments, looking for areas where the bases, the A, T, C, and G, line up. Once a match is found, a specialized protein called REC-A binds to the single-stranded ends of the broken DNA. REC-A guides the broken DNA strand to invade the matching DNA strand, kind of like solving a DNA jigsaw puzzle. Step 4. DNA synthesis. So, finally, the cell can start rebuilding. DNA polymerases, which are the enzymes that synthesize new DNA, begin copying the information from the intact strand into the broken one. This step restores the missing genetic information using the backup as a guide. Step 5. Ligation. Once the strand has been fully rebuilt, another enzyme called DNA ligase comes in and seals the repaired ends, making the DNA continuous again. This completes the repair. The DNA is now whole, correct, and ready to function like nothing ever happened. Fun fact. D. radiodurans doesn't do this just once. After a large dose of radiation, it performs hundreds to thousands of these repairs all over its genome in parallel.